Now, in part 4 of chapter 6, we are going to be talking about neurotransmitters and neuromodulators. These are chemicals that are made by neurons, and they transmit information from the neuron either to another neuron or an effector. And remember, we said that the effectors could be muscles or glands. And let's understand the difference between a neuromodulator and a neurotransmitter. Neurotransmitters, again, relay info from neuron to neuron or to the effector, while a neuromodulator is going to modify the postsynaptic cell's response to that neurotransmitter. So it can either enhance the response to the neurotransmitter or dampen the effect of that neurotransmitter. The neurotransmitter usually affects the cells through ion channels, okay, and that could lead to, again, if we open up sodium channels, we are exciting the nerve. If we open up other channels like chloride or potassium, we would be inhibiting the nerve. And the effect is very rapid, is very fast, happens within about, milliseconds. While neuromodulators are, on the other hand, their receptors are what are known as the metabotropic receptors. They lead to changes in the metabolic pathways of the cells. So they can increase or decrease protein synthesis. They lead to DNA transcription, enzyme activity, and these changes take a little bit more time. So these changes can occur over minutes or hours, or even days. So neurotransmitters are more involved in rapid communication, while neuromodulators are more involved in slower events, such as learning, development, and motivation. Now, these are the chemicals that we are going to be talking about during this portion of the chapter. So we'll talk about acetylcholine, biogenic amines, amino acids, neuropeptides, gases, purines, and lipids. So you can see here that actually biogenic amines, amino acids, and neuropeptides, these are all derivatives of amino acids one way or another. Okay, and we will, this list is obviously not all inclusive, that we have a lot of other neurochemicals that we are not talking about. These are the ones that are of the most clinical importance. So let's move on to the first one, and that is acetylcholine. Acetylcholine is one of the most major neurotransmitters. It is found in the peripheral nervous system and at the neuromuscular junction. So that junction, that synapse between nerve and muscle, the neurotransmitter in that synapse is acetylcholine. The receptors that are stimulated by acetylcholine are known as cholinergic neurons. The neurons that release acetylcholine are known as cholinergic neurons, while the receptors that are stimulated by acetylcholine are also known as cholinergic but receptors. Acetylcholine is synthesized by the combination of choline and acetyl-CoA. That happens in the cytoplasm of the neuron. And then when acetylcholine is released, we break it down by an enzyme known as acetylcholine esterase enzyme. That acetylcholine esterase enzyme is found in both the pre- and the postsynaptic membranes. Some chemical weapons, such as the nerve gas known as sarin, that inhibits acetylcholine esterase. And that leads to the buildup of acetylcholine in the neuromuscular junction, leading to the overstimulation of muscles. Okay, and that leads to uncontrolled muscle contractions, seizures, paralysis, spastic paralysis, and can lead to eventually to death due to, again, respiratory failure. Now we've got two different kinds of cholinergic receptors. They're namely nicotinic and muscarinic, and they are named according to what stimulates them. So nicotinic receptors are the same receptors that are stimulated by nicotine found in tobacco. Okay, these receptors are ligand-gated ion channels that allow the passage of sodium and potassium. We find nicotinic receptors at the neuromuscular junction and in the brain. And these receptors are really important in cognitive function and behavior. Okay, as you can see that whenever anybody smokes, they are stimulating these nicotinic receptors, and that could lead to kind of that shakiness that happens after smoking. It's also the reason why tobacco smoking is highly addictive, because we have a lot of these nicotinic receptors in the brain. And trying to withdraw from nicotine can lead to withdrawal symptoms. Now, the second kind of cholinergic receptors are muscarinic receptors, and it is named after muscarine, which is a toxin found in mushrooms. That muscarine toxin also stimulates those receptors, and the receptors are named after them. These receptors are G protein coupled receptors. They're found in the brain and in the parasympathetic fibers that affect the different parts of the body. We can block muscarinic receptors by an antagonist known as atropine, and we do that when we want to take a look at the retina of the eye. So when you take a look at the retina, you will be exposing the eye to a lot of light. And the normal reflex upon all of that light exposure would be to constrict the pupil. So you want to give something to pretty much paralyze the muscles, the pupillary constrictor muscles. So you give atropine. Okay, it is a muscarinic receptor antagonist, and it will dilate the pupil. Alzheimer's disease, which is dementia that happens, it is age-related. Okay, it affects about 10 to 15% of people around the age of 65, and 85 and better. At the age of 85 and better, about 50% of the population have some sort of Alzheimer's disease. It is due to the degeneration of the cholinergic neurons in the brain. So again, acetylcholine is really important for, memory. It is important for personality. It's important for language. And you will see that all of these things decline in Alzheimer's. So there is definitely memory loss. Okay, there is a decrease in language, so the ability to use words. There is a decrease in the cognitive abilities, so gradual decrease in the ability to read and write, to do math, and so on. There's also confusion. So obviously it's a spectrum, so it goes very gradually, towards the more severe cases. Okay, and that again is because of the loss or the degeneration of the cholinergic neurons in the brain. Now, the second kind of neurotransmitters slash neuromodulators that we'll be talking about are the biogenic amines. These are synthesized from amino acids. So we'll take an amino acid and change it up a little bit and make these biogenic amines. Okay, the most common ones that we have are dopamine, norepinephrine, serotonin, and histamine. So a lot of these terms and a lot of these chemicals are things that we, commonly have heard of already. We are now learning their role in the nervous system, which is pretty cool. Okay, epinephrine is also a biogenic amine, but although it is a neurotransmitter, but its more important function or more common function is it is the fact that it's a hormone that comes out of the adrenal medulla. So the adrenal medulla and the brain has and releases both norepinephrine and epinephrine. But norepinephrine is more of a neurotransmitter, while epinephrine is more of a hormone. And it helps that nor with an N is more of a neurotransmitter with an N, while epinephrine without the N is more of a hormone. Okay, so norepinephrine is an important neurotransmitter in the CNS and PNS. So you can want to think of them a kind of like neurotransmitter slash hormone, but each one kind of tends to go more towards one side of the aisle. these dopamine, norepinephrine, and epinephrine, that is one family known as the catecholamine family. It's made out of a ring called a catechol ring and an amine group, and we'll see the image in the next slide. These are all derived from the amino acid tyrosine. Okay, so we eat proteins, we break down those proteins. One of the amino acids is called tyrosine. Our bodies or our neurons will take that tyrosine and change it using an enzyme known as tyrosine hydroxylase to make these catecholamines. So I'll come back to this slide in a minute. But you can see here, this is the structure of the tyrosine. So you have your catechol ring. Okay, don't worry about the actual structure, but you do want to know these names, the catechol ring and an amine group. Okay, so which you can see here, your catechol ring and your amine group. Through the enzyme tyrosine hydroxylase, it'll change to L-DOPA, which is the precursor of dopamine. And dopamine will change into norepinephrine, which means that dopamine is the precursor of norepinephrine. These two in the darker grayish areas, these are your neurotransmitters. And in the adrenal medulla, norepinephrine is the precursor of epinephrine. Okay, so going back to this slide again, so all of these are part of the catecholamine families of three, the dopamine, norepinephrine, and epinephrine. We can break down these catecholamines by an enzyme known as MAO or monoamine oxidase enzyme. Okay, depression is a very important and prevalent disease, and there are different reasons for depression. And they're all kind of related to a lot of these neurotransmitters that we'll be talking about. So in some patients, their depression is due to the decrease in the levels of dopamine or norepinephrine. And one of the treatments that can be given to patients would be what are known as MAO inhibitors, so monoamine oxidase inhibitor. So you're basically inhibiting the enzyme that would break down or get rid of dopamine. And that would lead to the accumulation of dopamine in the body, or in the brain in particular, and that decreases their depressive symptoms. Let's know again that there's so many different neurotransmitters related to depression, so this might work with some patients and not others. Now, another biogenic amine is serotonin, and it is produced from an amino acid called tryptophan, while histamine is another biogenic amine, and its precursor is the amino acid histidine. So we already talked about the pathway of catecholamines, so we will move on to the receptors. So epinephrine and norepinephrine had an old name, which are adrenaline and noradrenaline. And that is why their receptors are known as adrenergic receptors. So the receptors kind of kept that old name. And we can find that, again, norepinephrine is more of the neurotransmitter, so it's found both in the CNS and the PNS, while epinephrine is found mainly in the peripheral nervous system. So again, it's mainly released by the adrenal medulla, and it's considered more of a hormone and is affiliated more with the peripheral nervous system. As I said, that the name of the receptors, adrenergic, really comes from the fact that their old names were adrenaline and noradrenaline. Okay, all of the adrenergic receptors are metabotropic, meaning that they are going to use a second messenger. And we have tons of adrenergic receptors. So these are the ones that you want to be familiar with. We have alpha adrenergic and beta adrenergic. In the alpha adrenergic family, there's alpha 1 and alpha 2, and there are lots more, but we are going to focus on alpha 1 and alpha 2. Alpha 1 are found in the postsynaptic neurons, and these are going to stimulate or inhibit potassium channels. So they are either going to, one way or another, are going to affect the, or change the behavior of potassium channels. Alpha 2 receptors are found in the presynaptic neurons, and that is to inhibit the release of more norepinephrine. So it's kind of like a local homeostatic mechanisms where norepinephrine comes out, and then the alpha-2 receptor is found in that same neuron, the presynaptic neuron that just released it, and that would lead to the inhibition or to decrease the release of even more norepinephrine. So it is kind of, again, like a local homeostatic mechanism going on. Beta adrenergic receptors, we have tons of those. So there's beta 1, beta 2, beta 3, and so on. And these are all stimulatory through the GS protein, and they all lead to the increase in cAMP in the postsynaptic cells. So you will find that, so why is it that we have all of these different receptors? Why do I care? How is this, in any way, shape, or form important to me clinically? Okay, the fact that epinephrine and norepinephrine have a wide range of effects is due to the fact that it has all of these different receptors. So for example, the effect on the heart would be stimulatory. So we have on the heart, we have these beta receptors, beta 1 receptors. Okay, and again, it will increase the cAMP, leading to the stimulation of the cardiac muscles. So the heart rate goes up, blood pressure goes up, and so on. Other effects, for instance, your norepinephrine and slash epinephrine also have stimulatory effects on the blood vessels, but it has inhibitory effects on the blood vessels. Okay, now you're really confusing us. So let's think. Epi/norepi is the sympathetic nervous system, okay, where you are in that stressful situation, flight, fright, fight mode. So you need the blood vessels, say to the kidneys, maybe, to constrict. Okay, this is not the time for the kidneys to keep on making urine when you are about to get eaten by that, proverbial bear that we keep on saying that is chasing us all the time. While on the other hand, you need the blood vessels that are going to the lungs to dilate in order to get more oxygen to the muscle so that you can run away from that bear. So adrenaline, or epinephrine, I should say, has two opposite effects on the exact same structure. So again, it can constrict some blood vessels and dilate other blood vessels due to the fact that it has all of these different kinds of receptors that respond to it. Now, next on our list is serotonin. Serotonin is also known as 5-hydroxytryptamine. It is a neuromodulator, okay, which means that its effect is going to be a lot slower. It takes its time to produce an effect. And it is found pretty much in every single neuron in the brain and spinal cord. And it has tons of different receptors, and we're not going to worry about those receptors, except maybe for the name of them if it comes up. Serotonin has different effects on the muscles versus senses. So on the muscles, it is excitatory to the muscles. Okay, so it excites the muscular system, while it inhibits senses. Besides the effect of serotonin on senses and on muscles, it also has an effect on regulation of food intake, reproductive and sexual behavior, and also on the emotional status. Serotonin is another hormone that is blamed for, or the lack of serotonin, I should say, is blamed for depressive symptoms as well. And you can see pretty much, from their effects, things that kind of reflect the depressive symptoms from, change in the food intake behavior. So either lack of appetite, loss of appetite, or increased appetite, change in sexual behavior, and changes obviously in the emotional status. So a lack of serotonin can lead to depression. An increase in the amount of serotonin can lead to anxiety. One of the drugs that are used to treat that is paroxetine. Paroxetine, again, is the pharmaceutical name, the generic name, while the commercial name, or the name of the actual drug that sold is Paxil. That is a SSRI. SSRI stands for Selective Serotonin Reuptake Inhibitor. So we will make sure that that serotonin remains over in the synaptic area, increasing the levels of serotonin, and that hoping that that would relieve the depressive symptoms. So again, depression has different neurotransmitters and neuromodulators that can lead to depression. So it is more of a trial and error with the patient. Try one medication. If it works, that's fine. If it doesn't, we move on to the next one. But then again, this could be very, annoying, very stressful to the patient. So you always want to keep that in mind and explain to the situation to the patient. You'll be surprised to know that the majority of serotonin is actually found in the digestive system. So about 90% of our body's total serotonin is in the digestive system. And that could kind of, when you think of it, it kind of makes sense because depression and anxiety, they are both related to, in a lot of patients, related to changes in the bowel habits. Okay, in the brain, we have about only 1 to 2% of the body's total serotonin is found in the brain. A drug that activates the same receptors is LSD, or lysergic acid diethylamide, also known, famously known as acid. Okay, these stimulate serotonin receptors in the brain, and due to that stimulation, they lead to visual hallucinations. Again, going back to, the function of serotonin, where it inhibits and the pathway that mediates senses, while it stimulates the pathways that lead to control of muscles. Now, the amino acid neurotransmitters. Okay, these are the most prevalent that are in the CNS. Notice that we are not including the PNS in this equation. So amino acid neurotransmitters are going to be found in the central nervous system, so brain and spinal cord. Some of them are excitatory, like aspartate and glutamate, also known as aspartic acid and glutamic acid, while the inhibitory neurotransmitters are glycine and GABA. We'll talk about the 3 Gs, so glutamate, glycine, and GABA. Glutamate is the first one, and that is the excitatory amino acid neurotransmitter. It stimulates 2 different kinds of glutamate receptors. Okay, they're known as AMPA and NMDA. You don't really have to worry about what these letters stand for, but you want to know you definitely need to know that the 2 kinds of ionotropic glutamate receptors are AMPA and NMDA. Okay, AMPA is, if you overstimulate AMPA receptors, that could lead to epileptic seizures. So some of the anti-epileptic medications block AMPA receptors. Okay, NMDA receptors, on the other hand, are related to plasticity of the brain. So remember we said that plasticity means the ability to form connections and to learn. So as we age, these NMDA receptors age as well and kind of disappear or decrease in number. So the thought is that if we can stimulate these NMDA receptors, that we will keep or prevent the loss of that ability. Okay, so again, as you know, as we age, it gets harder and harder to learn things, and that is due to the decrease in the number of NMDA receptors. So again, in order to keep that ability, we want to stimulate those receptors. And that the ability to learn is more of a cooperation between AMPA and NMDA receptors, and that leads to a process known as long-term potentiation. Long-term potentiation is the process involved in learning and memory, which basically means that we are wiring the brain to learn certain things. So for example, you are wiring that kindergartner that this letter right here, whenever you see that, that is an A, and this is how it's pronounced. When you see this letter, that is a P. So there has to be some kind of communication between these two receptors through glutamate, which again is a stimulatory neurotransmitter, and that leads to that long-term learning and long-term memory that we keep throughout life. Okay, so again, wiring the brain to know certain things. Here we'll talk about the more details on the long-term potentiation. So here is your presynaptic neuron, that is your postsynaptic cell, and that is the synaptic cleft. So when the action potential reaches the end of the presynaptic neuron, it will release glutamate. Glutamate will attach to these AMPA receptors. AMPA receptors are sodium ion receptors. So when we open those up, sodium enters into the postsynaptic cell. Okay, it leads to a little bit of depolarization, not enough, just a little tiny local graded potential. And we call that an EPSP, excitatory postsynaptic potential. It will not reach threshold, but because of that change of the potential, that opens up these NMDA receptors. NMDA receptors allow for the entrance of calcium. When calcium ions come in, calcium is going to act as a second messenger, and that is going to change the metabolism of the cell. It will lead to the synthesis of proteins, and you are literally changing the structure of that postsynaptic cell. So again, you are wiring these cells, making structural changes in the cells to learn different things. It takes a very long time, obviously. Okay, so the term potentiation means that you have to keep on, doing it over and over and over again. The potentiation also comes from the fact that the calcium that comes out here does a couple of things. Okay, so it increases the sensitivity of these AMPA receptors to to glutamate, and it also, we don't know how this happens, but it does. Okay, that it also increases the synthesis and release of glutamate from the presynaptic neuron, which translates clinically or translates in real life that the more you learn, the more you read the same information, you are learning it over and over and over again. Okay, and so reading something once, you will get some of the information, but if you read it 2, 3, 4, 5 more times and so on, you are increasing the amount of glutamate that is being made and the release, and activating these two receptors, leading to structural changes in the postsynaptic neuron. And these structural changes are the changes that we carry on for life. So now we know forever that that letter is a letter A. So these are the steps that we just talked about. So in step 1, your presynaptic neuron will fire off an action potential. It will release the glutamate. The glutamate will bind to the AMPA and NMDA receptors on the postsynaptic membrane. In the AMPA receptors, those, like I said, are sodium channels. They will lead to the influx of sodium into the postsynaptic neuron, leading to a local EPSP. When that happens, we'll open up the calcium ions channels, and that leads to calcium influx into the cell. Calcium acts as a second messenger in the postsynaptic cell, leading to the activation of different metabolic pathways, gene expression, protein synthesis, And it also increases the production and sensitivity of the postsynaptic neuron to glutamate. And somehow, by a mechanism that we do not know of, that calcium also increases the production and release of glutamate from the presynaptic neuron. So NMDA receptors are good, okay, except for the fact that they mediate something known as excitotoxicity. Excitotoxicity means that if somebody gets a stroke, okay, so a brain stroke that led to death of certain brain cells. Now those dead brain cells are going to release everything that's in those cells, including glutamate. So now there's a lot of glutamate in the extracellular fluid. That is going to lead to the overstimulation of AMPA and NMDA receptors. When that happens, that is going to lead to a lot of calcium getting into those healthy neighboring cells and leading to their rupture. Okay, so those stroked, dead neurons release again the glutamate, and that glutamate is going to overstimulate the NMDA and the AMPA receptors in the neighboring cells that are healthy. But because of the overstimulation, lots of calcium enter into these healthy cells, killing them, and then now these cells rupture, release their glutamate, and it's kind of like a domino effect, and that domino effect is known as excitotoxicity. So some of the new clinical trials that are out there suggest that if we give NMDA receptor antagonists, kind of block these receptors, would that prevent the spread of cell death that occurs following injuries to the brain? So these are different topics that you might want to research yourself, maybe ideas for your research project. That was your stimulatory neurotransmitter. Now we'll talk about the inhibitory ones. The first one is GABA. That is the major, it stands for gamma aminobutyric acid. That is something you want to be familiar with. It is the major inhibitory neurotransmitter found in the brain. And you will see that I've got things underlined, so you want to kind of pay attention to those. GABA neurons are found in the inner neurons. Remember, the inner neurons are the strictly found within the CNS only. And GABA can bind to either ionotropic or metabotropic receptors. Remember, ionotropic receptors open up channels, sodium, potassium, or chloride, or what have you. Metabotropic receptors lead to the activation of enzymes and protein synthesis. Okay. Because it is an inhibitory neurotransmitter, so you hopefully know that we are not going to open up sodium channels. We are going to open up chloride channels, and that flux of chloride, the negative chloride into the cell, is going to hyperpolarize the cell, leading to an IPSB. So you have definitely hyperpolarized, you have inhibited that cell. And we've got benzodiazepine drugs like Xanax and Valium. These act on the same receptors as GABA, and they can help to reduce anxiety. They can be used as anti-seizures, or we can use as to induce sleep. Okay, all of that due to the opening up of the chloride ion channels. Chloride goes into the cell, hyperpolarizing these cells and inhibiting them, okay, through the effect of these GABA receptors. The same thing happens with ethanol. So ethanol also stimulates these GABA receptors. And when you stimulate GABA receptors, you are inhibiting the cells. Remember, GABA is an inhibitory neurotransmitter. So there is this relationship between ethanol and GABA. Ethanol is going to stimulate, again, the GABA synapses, leading to the inhibition, and it also inhibits the glutamate. So not only are you increasing the effect of GABA, you are also decreasing the effect of glutamate. Remember, glutamate was excitatory. So as the blood alcohol levels go up, that leads to, inability to impair judgment, memory loss, loss of motor coordination, loss of balance, and so on. Doses of ethanol are way too high. That could lead to the suppression of the centers in the brainstem. And the brainstem has the centers for respiration and the cardiovascular center. So that could obviously be fatal. Now for glycine, which is the other inhibitory neurotransmitter. But this inhibitory neurotransmitter is found in the spinal cord and the brainstem. So remember that GABA was in the brain. Glycine is in the spinal cord and brainstem. It is also ionotropic, allowing for the opening of the chloride ions, leading to the hyperpolarization of the neurons. And its main function is to balance and regulate skeletal muscle contraction. So what actually happens is that the frontal motor cortex, which is the frontal part of the brain, sends out tons of messages to your skeletal muscles to contract. So I'm going to give out numbers. These numbers are not true, but just to illustrate what happens. So let's say your brain sends out 1 million messages to your muscles to contract. Your muscles are not going to respond to those 1 million messages. It'll respond, say, to only about 100 of them. Okay? And that leads to a very smooth muscle contraction. Had your muscle contracted and responded to those 1 million messages, we would pretty much be rigid, and we would have what is known as a spastic contraction, pretty much no relaxation. So that inhibition, that removal of all of those extra unneeded motor impulses coming from the brain is due to the effect of glycine. So glycine will regulate your skeletal muscle contraction, okay? It will get rid of all of those unnecessary, unneeded, and extra signals going to your muscles. So when we lose that, okay, for example, in the case of poisoning with strychnine, which is found in some pesticides, strychnine is an antagonist to glycine receptors. So you lose that inhibitory effect. Now, those 1 million messages are going to now reach your skeletal muscles. Those skeletal muscles are going to maybe go through a seizure, could go through spastic contractions, pretty much just contraction without any kind of relaxation. And again, the danger of that would be when it affects the respiratory muscles. Now for neuropeptides, these are a little bit bigger in size than the ones that we just talked about. So these are made out of 2 or more amino acids linked together to make a peptide. And we have tons of them that we are not going to really discuss in detail. But what happens is that they are made in the cell body and they go down the axon, if you guys remember those little cute videos that we watched, and they are going to be stored in the vesicles at the axon terminal. Okay, when they are released into the synaptic cleft, we break them down through these specific peptidases, which are enzymes to break down peptides. And any neuron that releases neuropeptides is known as a peptidergic neuron. Same goes for the receptors, they have the same names. Most of the times, these neuropeptides are actually neuromodulators. So they modulate the response to neurotransmitters. Now, these are, without going into detail, these are the examples of neuropeptides that we want to know. Like I said, we've got hundreds of those. We only need to know 3. So they are known as the endogenous opioids. We have in our body, we have an internal source of opium. Okay, they are known as beta endorphin, dynorphins, and enkephalins. These are mood enhancers, so they make us feel better. mood enhancers and mood stabilizers. They decrease also in depression, leading to depressive symptoms. So again, you see that a lot of these different neurotransmitters and neuropeptides are really the cause or can lead to depression. And that's again why depression is such a hard disease to treat, because it could be, there's so many different reasons leading to it. Opioids are also, or these endogenous opioids, not only do they stabilize the mood and enhance our mood, they also decrease our perception of pain. Okay, so you can also see where if a person is depressed, they now feel more pain. Okay, so it's kind of like a cycle where the depression leads to more pain, and pain leads to more depression, and so on. And there's a cycle that needs to be broken at some point. One of the ways to break that would be, obviously the antidepressive medications, a couple of which we've already talked about, or painkillers. Okay, so painkillers, if you break the cycle through getting rid of the pain, well, maybe the depression will get better as well. Again, the patient really does not really care about this philosophical discussion that we're having, but it is really important to know that maybe the cause of the depression is really the pain that they're feeling. Although the pain is not due to a physical disease, meaning the pain is very true, but it's not due to, say, arthritis or an injury. Okay, so there's nothing really pathologically wrong to treat, but it is due to the changes in the levels of these neuropeptides, the decrease of the levels of these neuropeptides. So it's much harder to treat than arthritis. Another time when these beta endorphins are low would be right before the premenstrual period. So the PMS, or premenstrual syndrome, which is, in some women, go through a little bit of, depressed mood or just feeling down, bloating, a little bit of pain that happened right before menses. Well, that is due to the decrease in the level of these endogenous opioids. And one of the things that is very rich in beta endorphin is dark chocolate. So a lot of the times, women kind of gravitate towards chocolate right before their periods. We also have synthetic opioids. So these are synthetic. We do not make them, okay? Or we don't make them internally, at least in the body. So morphine and codeine, those are used as analgesics. So pain reducers, okay? Because they have the same action and they stimulate the same receptors as the endogenous opioids do. Some neurotransmitters are actually gases, which is really interesting because these gases are also made in the cell body. They are going to be released from the presynaptic vesicle through exocytosis, and they diffuse to the surrounding cells. Examples of these gases are nitric oxide, carbon monoxide, and hydrogen sulfide. Nitric oxide is a vasodilator, okay, which means, and it is found in blood vessels. So one of the ways our blood vessels can dilate would be by the production of nitric oxide. Now, purines, so ATP and adenosine, we really think of ATP as a source of energy, but it is also a neuromodulator together with adenosine. Again, they're found in the presynaptic vesicles. They are released with other neurotransmitters. And adenosine is really derived from ATP. And they, without going into the details, okay, they just know that they play a role within the nervous system. And it is something that we really don't know a lot about. There's still a lot of research going on about it, so not much to know here. Lipids, that we kind of talked about the eicosanoid family of molecules that was derived out of the arachidonic acid in the case of whether inflammation or allergies. These are neuromodulators. They stimulate a group of receptors known as cannabinoid receptors. And yes, the name comes from cannabis because cannabis stimulates these exact same receptors. And they are found, these receptors are pretty much widespread throughout the CNS and the PNS. They have a wide range of function, including effects on the appetite, pain, mood, memory, and muscle activity. Like I said, the receptors are the same target of cannabis, and that's why when they're activated, the cannabis leads to these different, that range of effects. So at this point, we are going to end this portion, and I will see you in part 5.